Hi everybody. In this video we're going to go through the endocrine system. So this has to do with hormones and how they are used to control the internal environment of animals. So just to kind of review the different types of intracellular signaling that occur, in A you can really see endocrine signaling, which is what we're going to talk about here, in which case specific endocrine glands are releasing signals, hormones in this case, into the bloodstream, and they're going to travel and pop out and then trigger a target response in some kind of target cell. Paracrine signaling has kind of a similar approach, although para means next to, so these are really local regulation responses, in which case the signal molecule is released into a very close environment, not necessarily the bloodstream. Autocrine has to do with self. So in essence, the release of molecules that are signaling from the cell can actually trigger the receptors of that same cell. We can also talk about neuron signaling or synaptic signaling, and this is when neurons are talking to each other. And then finally, which we'll circle back to in this specific PowerPoint as well, is neuroendocrine signaling. So there's some kind of neural signal that will trigger endocrine release into the bloodstream and then have a downstream response. So in terms of some general vocabulary, the endocrine system is the whole body system that incorporates the cells and all the tissues that are associated with it. That consists of endocrine glands, which are ductless, secreting hormones into body fluids. Usually, in this case, it's going to be the blood. The hormones themselves are going to act as the signal that cause some kind of response. So whether those hormones are triggering G protein coupled receptor signaling, or those hormones could be opening ligand gated ion channels. They could also be doing intracellular signaling. They're acting as the ligand. They could affect one tissue depending on the target. They could actually affect more than one. Oftentimes it's also possible that depending on the signal, they could affect most tissues in the body. And then in general, just understand that when we're talking about cell communication, all of this is regulated by feedback. So whether it's a positive feedback or a negative feedback, and I'll show you examples of that. Highlighting hormones in the middle, just kind of covering different types of signaling molecules. The hormones are the ones that go through the blood, and this is going to include peptides and steroids. It's a little bit different than other things, like a pheromone, for example. That term, just to kind of make it clear on the left, is something that's secreted, usually by like a specialized skin cell, but to talk to one specific individual to another. So ants do this so that they all follow in a line. Bees have like sex pheromones that they give off. Whereas on the right, we have local regulators. These are signals that are secreted by cells that are near each other, whether it's growth factors or cytokines, which we'll talk about later, or nitric oxide, those kinds of things. We're going to be mainly focused on the middle. This picture just shows you what the endocrine glands are. So it's not something that we always cover in every class, but the main heavy hitter is the pituitary, and you can kind of get an idea of all of the different hormones that are released from the pituitary, specifically the anterior pituitary. It's a lot. The hypothalamus, we'll learn, will control the pituitary. But we also have other glands like the thyroid, parathyroid, your adrenal glands, which has two parts, the medulla and the cortex. Your ovaries and testes are considered endocrine glands because they produce estrogen as well as testosterone. And androgen is a testosterone. And then your pancreas. I feel like that's always talked about a lot in terms of insulin production and diabetes. So this is an example of a simple endocrine pathway. And as I mentioned on the AP test, they're really going to be wanting to show you a lot of pathways. So a simple system is there's some kind of stimulus, and that is going to trigger an endocrine cell to release the hormone into the bloodstream, and that bloodstream is going to have that signal pop out for a target cell and have a response. And a lot of times in this case, it's showing you negative feedback. That specific response is going to then negatively impact or go back towards the medium homeostasis and negatively impact the initial signaling. One specific example seen on the right is secretin signaling, and this has to do with pH levels. So an initial stimulus might be that the pH has gotten too low in the duodenum, or duodenum, however you say it. In this case, then, that low pH is going to trigger specific cells. Those cells are going to release secretin. Secretin is the hormone in this case. Secretin will then travel in the bloodstream and target pancreatic cells. Those pancreatic cells are then going to release or trigger the release of biocarbonate. And biocarbonate is a base. So by releasing biocarbonate, it in essence is going to raise the pH or negatively impact the low pH that was happening. So again, it brings it back towards that set point we've learned about previously. An example of a positive feedback would be related to oxytocin signaling. So on the left, you can see a general pathway here where there's a stimulus, and then that triggers a neurosecretory cell or some kind of secretion of the hormone into the bloodstream. And then it will travel to its target cell and cause a response. And then that response positively impacts the stimulus. So then you're going to end up actually making more and more and more and going farther away from the set point. So oxytocin is released by nursing mothers. So a stimulus for this is suckling, and that suckling action causes the release of oxytocin, and that hormone causes the smooth muscle or targets the smooth muscle in the mammary glands that will trigger milk release. So the more suckling, the more oxytocin release, the more milk release. And then it's going to positively impact more and more and more. For example, in this case, for a nursing mom to feed her child. This slide and the next slide are just going to kind of cover what the major hormones are. I'm not saying you have to memorize this table for sure, but it just gives you an idea of what the different hormones are and generally what they do. And it also tells you where they come from. So for example, the thyroid gland releases T3 and T4, and those are specific hormones that help to regulate metabolism. Or there's also, like in the last slide, we had oxytocin. It's released by the pituitary gland, and it stimulates uterine contractions and mammary gland cells. On this slide, just one general example that usually gets talked about a lot, I could say the adrenal medulla, which is part of the adrenal gland, releases epinephrine, and that's really in charge of the fight or flight response. It also can raise blood glucose levels and a bunch of other things. We mentioned negative feedback and positive feedback. Here are just some specific examples of where those would fit in. So thyroid hormones regulate metabolism, so it's important that you maintain that set point. And so there's a negative feedback involved. The downstream signals feedback to the original. Blood calcium levels also need to be tightly regulated, so that's another example. And blood glucose levels need to be heavily regulated. Whereas, as I previously mentioned, oxytocin is a positive feedback. In certain circumstances, you want to be heavily away from that set point after you have a child. Okay, so this has been alluded to in the past, but just to kind of go through what this is, there are two major types of hormones. There's peptide hormones and steroid hormones. Peptides are water soluble, so that means that they cannot just pass through the plasma membrane on whatever target cell they want. So they need to actually bind to the receptors in the plasma membrane and trigger a transduction pathway. So this includes the G protein coupled receptors, GPCRs, or the ligand-gated ion channels. And so they're going to affect proteins that are already there in the cell. They can either turn on or off those particular ones that are already in the cell. It's a rapid response because the proteins are already there, but usually it's a short-lived thing. You're not specifically affecting gene production, so it's more of a short-lived thing. And this includes oxytocin, insulin, and epinephrine. Some of those things you wouldn't want to be a long-lived response anyway. Whereas a steroid hormone, steroids are lipid soluble, so they can actually enter the cell, and they bind to intracellular receptors, which we talked about before. They cause a change in gene expression, so they have a longer lasting change because they're literally turning genes on or off. Therefore, it's slow. It takes some time to turn these things on or off. So because of that, it's a slower response. Testosterone, estrogen, and several others are steroid related. Here's a picture of what that might look like. So on the left, you can see peptide hormone. On the right, you can see steroid hormone. The peptide one you can see goes through the bloodstream and has to bind to a receptor in the membrane, and it can immediately target cytoplasmic proteins, or it can potentially, eventually make its way and do some gene regulation. Whereas the lipid-soluble ones can automatically, they themselves go through the membrane and to an intracellular receptor and then can affect gene regulation and eventually trigger a cytoplasmic response. It's important to know that in a lot of these signaling cascades, we have, it seems like a lot, but we don't have a lot of hormones in general. And those few hormones can have a variety of responses. So for example, on the left, we have a liver cell, and all of these are related to epinephrine, again, the fight or flight hormone. And so one of the necessary responses in the fight or flight response is the liver cells releasing a bunch of glucose. So epinephrine will bind to receptors on liver cells, and those liver cells are holding on to glycogen deposits, which can be broken down into glucose. So it triggers the cell to break down glycogen and release and dump a whole bunch of glucose outside the cell so that it can be used as energy. Additionally, epinephrine at the same time is going to increase blood flow to the skeletal muscle. And so it does this by binding to receptors on smooth muscle and cause it to relax. And then that will dilate the blood vessels and allow blood to flow a little bit more freely. On the same token, blood or smooth muscle cells in the intestines want to constrict because you don't really want to worry about digesting food in a fight or flight response. So they're going to bind to alpha cells, which are different than beta cells, but it's still a receptor. And they're going to bind and actually cause the cell to contract. And then that will cause blood vessels in that area to constrict. So this is kind of highlighting the idea that one specific hormone can have a myriad of effects depending on which target cell and receptor it's binding to. Now you can see roughly kind of one of the master control center parts of the brain is called the hypothalamus. And the hypothalamus often triggers the pituitary gland and tells the pituitary gland to turn on and release specific hormones. That's why it's really called a master gland. And so it's going to receive information from the nerves in the brain, initiate endocrine signals. And here you can roughly see what some of the main hormones are that are released from the posterior pituitary and the anterior pituitary. In terms of the posterior pituitary, you can just see there's a number of different hormones. Here we have ADH, which is antidiuretic hormone, as well as oxytocin that we had been talking about before. The anterior pituitary has a host of specific hormones that are released for various different reasons. You don't have to memorize exactly which hormone comes from which endocrine gland and does exactly which thing. Most of the time you're going to be probably given a scenario where they tell you this information and then you just have to use it. So here's an example of one of those pathways. In this case, it has to do with blood glucose levels. So maintaining homeostasis is important, so the set point of blood glucose is kind of in the middle. And what happens was when your blood sugar rises, let's say you ate, you did carb loading for sports, so you ate a giant bowl of pasta. What's going to happen now is that the beta cells of your pancreas are going to release the hormone insulin into the blood. And then what happens there is then that insulin hormone causes your body cells to take up more glucose to kind of clear it away. And those liver cells take up and store it as glycogen that can later be broken down if needed. And because you're removing the glucose, your blood glucose levels go down back to the set point. However, if you haven't eaten for a while, your blood glucose level might go down, and that triggers alpha cells to release glucagon. And the glucagon that has been stored up can be broken down and released as glucose so that your glucose in the blood rises back to the set point. So again, these are negative feedback loops because you're really trying to, when something goes one way or the other, get it back to that major set point. So you're counteracting what was there. And this graphic in your PowerPoint just kind of, again, highlights that idea of what blood glucose control is doing. Now, when that gets out of hand, we end up with disorders. So here, diabetes mellitus is the diabetes that we know related to insulin. There's also diabetes insipidus, which is related to something else. Type 1 and type 2 diabetes are a little bit different. Type 1 diabetes is the one that, people are essentially born with. That's about 10% of all diabetes cases. And it's called insulin dependent because what happens is the beta cells of the pancreas get destroyed. So they are no longer, the endocrine cells essentially are no longer able to release the hormone insulin. And that's a problem because then you can't release insulin. If we remember back to going all the way back to our picture, if the beta cells can't release insulin into the blood, then they are unable to make blood glucose levels decline because the insulin in the blood will trigger the uptake of glucose. And so you're going to continue to have high glucose levels in the blood. Whereas type 2 diabetes affects 90% of the population, and that's really a receptor problem. It's called non-insulin dependent because it really doesn't depend on the production of insulin. Your beta cells are still functioning properly and releasing insulin. The problem is that the target cells don't actually respond to it. So insulin is made, but the cells don't respond. So this is a receptor signaling problem. Obesity and lack of exercise are examples of risk factors that could lead to that. Another example pathway here is the thyroid gland. So the thyroid regulates metabolism. So in this case, we have, in step 1, there's some kind of lowering of thyroid hormone levels. There, you receive the signal that it has dropped. So the neurosecretory signals, again, this is a neurosecretion because the neural signaling is telling you that it's lowered. So it's going to secrete TRH into the blood. And that particular hormone is going to travel to the anterior pituitary and cause the release of TSH. So in this case, the neurosecretory cells are secreting and triggering the pituitary to release a hormone, TSH, that will go into the blood and then eventually make its way to stimulate the endocrine cells in the thyroid to release T3 and T4. This particular pathway, you can notice, is really a pathway of multiple hormones being released in kind of a domino effect. So once the thyroid releases T3 and T4, then it can actually go through and trigger a response. So it can act in a negative feedback capacity because T3 and T4 originally increase in the blood and act on target cells. But once they do that and they return to a normal range, then those hormones can actually block the further release at multiple points. So it can block the TRH release and it can also block the TSH release so that it goes back to normal levels. And then when they drop to a significantly low portion, then the feedback isn't there anymore, and then you can make more of this process. One example here, just to kind of highlight, is Graves' disease. It's another cell signaling problem. It's an autoimmune disorder. Antibodies bind to the TSH receptor, so this is related to the thyroid gland, essentially, and can trigger something called hyperthyroidism. So this result will trigger, normally you have the TSH triggering the TSH receptor and stimulating hormone release from the thyroid cell, and then you regulate your metabolism, and it's nicely negatively feedback controlled, and so on and so forth. However, in Graves' disease, it's an autoimmune disorder, so the antibody is targeting the thyroid cells. It's going to bind to the TSH receptor so that the TSH itself can't bind, but it's going to essentially force the cells to stay on and release all of this all of these regulated signaling molecules released from the thyroid cell, and then you get too many thyroid hormones produced and your metabolism is out of control. So that's why it's called hyperthyroidism. And it becomes unregulated and not able to be negatively feedbacked. So you can lead to things like sweating and weight loss because your metabolism is ramped up. It can give you high blood pressure and a number of other things. Thyroiditis, aka a goiter, is another example of one thing that can happen. So in this case, you have an autoimmune destruction of the thyroid follicles. So the thyroid cells themselves are not functioning as they should. So what that means is you are releasing less T3 and T4 than you normally would. So you have a chronically underactive thyroid release. And because of that, then you're not really going to get any negative feedback. So it increases your TSH levels to try to compensate. So it's a little strange, but the lack of work by this guy down here in the thyroid gland causes a lack of negative feedback. And so you're producing a lot of TSH as a result. But these last few slides are just really an example of what those things are. So just mostly be prepared to look at signaling pathways and identify how things can change if you modify it slightly. The end.